Okay, so good evening, everybody. Welcome to the the penultimate session for classics in neuroscience. Uh, yeah, I'll just share my screen. That's better. As I said, it is the penultimate session, as in there'll be just one more session after this for this particular course, Classics and Neuroscience. In this week's lecture, you might have studied about a couple of other new Nobel laureates and how they have, what were the particular experiments they have done, which we have covered in this extra session class earlier itself. What we were doing is, apart from that particular week's previous year questions, we were also trying to understand how learning and memory that is occurring. And in that, for that, it is the final session. So we'll wrap up how learning and memory is occurring. That bit also we'll wrap up today. So that, before we take up that part again, let us quickly begin with previous year questions for week 11, right? So you all know how the format is going to be by now. So we'll quickly go through the questions. So if we look at question number one, it is, what did Bernard Cart's research primarily focus on? So the options given were brain development, neurotransmitter release in quanta, human memory processes, or muscle contraction. So which one of these do you recall as being the primary focus of Bernard Cart's research? Right? So it should be very fresh in your memory if you've gone through Sir's lectures this week. And you'll probably know already that it was primarily focusing on the release of neurotransmitter in quanta, right? So he was working, this is his Nobel laureates page, that fact screenshot. So this is Bernard Katz. He won a Nobel Prize in Physiology or Medicine in 1970. He was born in Germany. He died while he was at UK, affiliated to University College London at UK. And the motivation for his prize that he shared with two other scientists was for their discoveries concerning the humoral transmitters in the nerve terminals and the mechanism for their storage, release, and inactivation. These transmitters are basically neurotransmitters, right? So, moving ahead, there are more than a couple of questions. So I think four or five questions based on Cards and his research. So we'll be discussing the research also very soon. So the second question is, which neurotransmitter did Cards research focus on at the neuromuscular junction, right? Was it dopamine, serotonin, acetylcholine, or glutamate, right? So, if you recall, this was actually one of the first studied neurotransmitters, much before these other ones, that is dopamine, serotonin, and glutamate, had been studied. This neurotransmitter was acetylcholine, right? So, acetylcholine is abundantly found as a neurotransmitter responsible for muscle contractions and this transmission of the neural impulse to the muscle, right? Dopamine and serotonin are present in the brain. Dopamine is largely involved in the reward system of the brain. Serotonin is what we know as the happy hormone. And glutamate is another excitatory neurotransmitter that is abundantly present in the brain, right? Apart from some PNS these junctions also. So, if we read more about Bernard Cart's work that was mentioned in this Nobel Prize, it says the nervous systems of peoples and animal consists of many nerve cells with long extensions of nerve fibers. Signals are conveyed between cells by small electrical currents and by special substances known as signal substances. Right? What are these signal substances? We now know them as neurotransmitters. So the transfers occur via contacts or synapses. In 1950s, Sir Bernard Katz studied how impulses in motor neurons activate muscular activity by measuring variations in the electrical charges. For example, what he showed was how the signal substance acetylcholine in synapse is released in certain amounts, right? So, which was the neurotransmitter he was working at? You are getting a clear answer for that also over here. Also, the story of how neurotransmitters are stored, released, and inactivated within such a short space of time is told by the achievements rewarded with 1970 Nobel Prize of in physiology or medicine. In this contribution that was particularly for Bernard Katz, this is an extension of the Nobel summary, right? So that is what has been listed over here. So Sir Bernard Katz used a microscopic recording electrodes to measure the electrical changes that occur when the chemical transmitter acetylcholine is released by the nerve cells, right? So once this neurotransmitter is released, what he found was from a series of measurements, he deduced that acetylcholine is released in highly defined amounts, right? So it will not be like abundance of acetylcholine, very small acid, volume of acetylcholine getting released. It will be a very, very fine-tuned amount. And that acetylcholine molecules are stored in small bubble-like compartments or synaptic vesicles in nerve endings. When an impulse arrives, millions of acetylcholine molecules are released into the synapse almost immediately. And Katz showed that calcium plays a key role in triggering this so-called quantal release of acetylcholine. Right? So this was what the first question was based on. Thereafter, another question on Cart's experiments was, what was the key experimental observation which supported Cart's quantal release hypothesis? Was it the spontaneous miniature end plate potentials or nerve impulses crossing the synapse, or the increase of neurotransmitter release with increased calcium concentration, or continuous release of neurotransmitters, right? So this was actually a work done by Katz in collaboration with another scientist. This other scientist, uh, we'll discuss about shortly. The answer for this question is spontaneous miniature end plate potentials, which are also known as MEPPs, right? So Fat was this other scientist. Both of them basically obtained the first clue as to the quantal nature of synaptic transmission in 1951. What they did was they observed spontaneous postsynaptic potentials of approximately zero point 0.5 millivolt in the nerve muscle synapse of the frog, like end plate potentials evoked by nerve stimulation. These small depolarizing responses were largest at the site of nerve muscle contact and decay electrotonically with distance. So this is like any electrical signal, right? So where it is produced, or any signal for that matter also, right? Even in case of earthquakes, you see wherever is that focal point, the maximum magnitude, as the focus is as we move away from the focal point, it keeps dampening, the signal keeps dampening, right? It happens with any kind of signal for that matter. So this was that experiment, right? The amplitude of the postsynaptic potential basically decreased with time, and course of potential slows with distance from the site of initiation in the end plate, right? So if this was the actual region, uh, if this was the actual region where the nerve was making contact with the muscle, here this potential will be the highest, as you can see, and as we move away from this region, the potential will keep decreasing, right? Also, what was seen was uh, this other response, that is, this is the motor neuron terminal, and you can see that the decay which we saw in the earlier schematic or representation, it results from leakiness of the muscle fiber membrane, right? So if we are over here, it has only leaked this much. If we are over here, it has leaked over here, here, right? So that is why it keeps getting diluted, because there is a lot of leakage that is constantly occurring, right? So, for instance, you take a wire and you don't insulate it, right? But the current you apply at one end will not be the same you receive at the other end, right? That is how it works, basically. Because charge must flow in a complete circuit, the inward synaptic current at the end plate gives rise to return outward current through resting channels and across the membrane, which acts as a capacitor in this case. This return outward flow of positive charge, as you can see over here, it depolarizes the membrane because the currents leak out along the membrane, and the outward current decreases the distance from the end plate, right? So this is what it's being talked about, the synaptic currents. So, post this, small spontaneous potentials have since been observed in mammalian muscle cells and in central neurons also. Because the postsynaptic potentials at vertebrate nerve muscle synapses are called end plate potentials, Fat and Kurtz called these spontaneous potentials as miniature end plate potentials. Several results convinced Fat and Katz that the miniature end plate potentials represented responses to the release of small amounts of acetylcholine and the neurotransmitter used at, which was the neurotransmitter used at that NMJ, right, neuromuscular junction. Then comes question number 4, which was what did calcium ions play, what role did calcium ions play in neurotransmitter release according to Kurtz, right? So option A was it prevented neurotransmitter release, or it triggered neurotransmitter vesicle fusion, or C, that is increased the vesicle density, or D, which is stability transmitter molecules, right? So calcium ion in general, whenever you study it, in case of the neurotransmitter, most places you will always see that calcium efflux is basically triggering a response, right? So over here also that is true. So B is the correct answer, that is triggering neurotransmitter vesicle fusion, right? So calcium influx is actually responsible for a lot of tasks that happen in the body at a molecular level, right? So that is why that SCR is present, which has a pool for calcium, which when it gets triggered, lots of calcium are released simultaneously. Contraction of muscle is also a response of that only, right? So generally it triggers responses. So over here, Katz and Milady next turned their attention to calcium ions, right? Post that work on the synaptic quantil, and then about this thing, um, miniature and plate potentials that we were studying. Post this, they turned their attention to calcium ions, right? Earlier, Katz and Jose del Castello had found that increasing the extracellular calcium concentration enhanced the neurotransmitter release, whereas lowering this concentration reduced and ultimately blocked the synaptic transmission. Because the neurotransmitter release is an intracellular process, these findings implied that calcium must enter the cell to influence the transmitter release, right? So, Katz and Milady found that calcium channels were much more abundant at the presynaptic terminal, right? So this is where the fusion has to happen with the terminal of this presynaptic neuron, so that vesicles can fuse to this terminal and neurotransmitter can be released, so that postsynaptic neuron can actually take that signal and carry forward that impulse, right? So there in the presence of tetraethyl ammonium and tetrodotoxin, a depolarizing current pulse was sometimes able to trigger a regenerative depolarization that required extracellular calcium or a calcium spike, right? So ethyl ammonium and tetrodotoxin, both of these we have studied earlier. One of them blocks the potassium ion channels and the other blocks the sodium ion channels, right? So what they are saying is even when these compounds are applied, right? Even despite that, when sodium or potassium ion channels might not be functioning at all, sometimes we can still elicit a small response if a calcium spike is given, right? So Katz and Milady therefore proposed that calcium serves dual functions, right? So one is that it is a carrier of depolarizing charge during the action potential like sodium, and it is a special chemical signal, a second messenger conveying information about changes in the membrane potential to the intracellular machinery responsible for the neurotransmitter release, right? Then we come to question number 5, which says, which experiment provided evidence for the quantum release hypothesis, right? Was it the measuring electrical brain waves in humans, or recording miniature end-plate potentials in neuromuscular junctions, or examining neurotransmitter degradation in the brain, or observing synaptic vesicles under the electron microscopy, right? So which one do you think will be the correct answer? I can, so you can safely rule out the first one, right? Brainwave pattern has nothing to do with the directly associating how quantum release patterns are happening, right? So option B is actually correct, which was recording these miniature end-plate potentials that we just studied about at NMJs. They provided evidence for quantum release hypothesis of these cards, right? Then we come to another Nobel Prize winning scientist, Roger Sperry. Um, I'm sure you have also gone through his work this week. Just give me a moment. Yes, we'll reshare the screen and we can begin the discussion again. Yeah. Yes. So what was actually the focus of Roger Sperry's Nobel-winning research, right? Was it the role of neurotransmitters in the brain, or the effects of severing the corpus callosum and hemispheric specialization, or brain growth in infants, or the electrical signals in the heart, right? So Rogers Pelletri was actually very famous for his work with split brain patients. So that is why this corpus callosum one is the correct answer. That is the effects of severing corpus callosum. That is, if the corpus callosum, which is the strip that is basically joining the two hemispheres of the brain, the right and left, if that is damaged, then the right and left hemisphere of the brain can no longer interact with one another, right? So what effects does that kind of a thing causes, right? So that the patients in which this kind of a condition happens is known as are known as split brain patients, and they are the subjects of Roger Perry's work, right? Basically, his work focused on those patients. So this is Roger Perry. He basically received Nobel Prize in Physiology or Medicine in 1981. He was born in USA, Hartford, and he died at Pasadena at Caltech, USA. And the motivation for his prize was for his discoveries concerning the functional specialization of the cerebral hemispheres. And interestingly, although it says here that the prize share for Roger Sperry was 1/2, he actually shared the prize with three other people. So, because it was understood that his contribution to this field was more than them, he was given half the prize, and the two of them were given one fourth of the prize each. So, this is one of the rare cases that also happens, right? Uh, so what did Sperry discover about the left hemisphere of the brain, right? So he made some discovery about the right hemisphere, some about the left hemisphere. So what was it that he discovered about the left hemisphere of the brain? One, it controls emotions. B, it is primarily responsible for language and logic. C, it is involved in the visual processing. Or D, it manages spatial awareness, right? So, which one of these do you think was the correct one for his discovery about left hemisphere, right? So, Sperry basically discovered that left hemisphere of brain is responsible for language and logic, right? What exactly he stated was that both human and animal brains have two halves with somewhat different functions that we know. One way Roger Sperry studied these functions was by examining patients whose hemispheric connecting nerves had been severed to alleviate serious epilepsy. So over here, it had been done um, through a surgical procedure, right? So it might have been possible that there were people who were epileptic, and their focal point of epilepsy might have been in the corpus callosum or near one focal end in the brain, but that focal end was also sending signals to the other region of the brain. So it was getting, spread out so much that they thought that if we sever this connection, what will happen is it will keep restricted to this region, and the other region of the brain will no longer get affected, right? So what he saw was that by 1960s, he could reveal that left hemisphere is more geared towards abstract and analytical thought, calculation and linguistic ability, right? So not just language and logic, but also calculation, analytical thought, and abstract thought, right? While the right hemisphere is more responsible for comprehending spatial patterns and complex sounds like music. So it is also a common saying that people who are right-handed are more, studious, analytical, logical people, and the people who are left-handed are more creative, and expressive, creative, and artistic people. So that arises from this logic itself, right? So the right uh, side of our body is actually controlled by the left side of our brain, and the left side of the body is controlled by the right side of the brain. So this was Perry's statement through which that logic then stems, right? Okay. Then we come to question number 8, which says, what concept did Sperry's split brain research lead to regarding consciousness? So, what did he tell us about consciousness? One, consciousness is a unified process in the brain. B, consciousness arises from the interaction of two independent cognitive systems. C, consciousness is controlled by the left hemisphere alone. Or D, consciousness is irrelevant to the brain function. So definitely not D, right? B is actually the correct option, that it arises from interaction of two independent cognitive systems in the brain, the right part of the brain and the left part of the brain. So, question number 9, in Sperry's research, which hemisphere is responsible for spatial awareness and visual processing? We just read about it, right? So, is it left hemisphere, right hemisphere, both hemispheres equally, or neither of these are hemispheres, right? So, we know it is right hemisphere now. Then, question number 10 is, what did Sperry's experiments reveal about the communication between spheres in split brain patients, right? A, the hemispheres continued to communicate normally. B, the left hemisphere controlled both sides of the body. C, the right hemisphere could verbalize stimuli from left visual field. Or D, the hemispheres functioned independently without direct communication. Right? So, option D is actually the correct option because in case of split brain patients, the connections that were interconnecting these two brain hemispheres were lost, because of which the two brain regions were independently, functioning. So there is this brain by V.S. Ramakrishnan. I am not sure. The name of this book is. Okay, now I have forgotten everything. Mm, Okay, maybe I'll just attach a link. So there is a this book by a neuroscientist, a neurosurgeon. At that, he's an Indian origin neurosurgeon in US, and in one of his chapters, he has also talked about the patients who had this kind of a split brain situation. Although it was not surgically induced, it was there by birth, due to which what the left part of the brain thought was very different from what the right part of the brain thought. And one part of the brain was actually self-lethal. So, like this woman who did have this condition, it used to report to her doctors that it feels like my left hand is trying to, suffocate me or kill me. And it was actually and it was thought naturally that the woman is hallucinating. She was sent to psychologists. She was sent to multiple neuroscientists, and this was the explanation that this particular doctor gave, but it could not be tested at that time, right? Few months or few years later, once this woman died, the brain was actually autopsied, and this was actually proven to be true, that actually the corpus callosum was missing in the woman, and that is why one part of the brain uh, was like joyous, cheerful, everything, and the other was kind of suicidal, and both of them she had kind of a split personality because of that also, right? This was the exact experiment that Rogers Perry did. Um, Over here, you can see in the, okay, we have also mentioned this before, right? We have studied this experiment before, but just for a recap of some sort, we can understand this again. So, in the normal condition, this is how the visual fields look like in case of the uh, frog over here, right? And in this case, it is color coded. As you can see, it is red, yellow, green, blue, right? And red has this connection to the posterior end. Green has extension to the anterior end. Similarly, the yellow has the extension to the ventral end of the brain. And blue has extension to the dorsal end of the brain, right? So there is some kind of a proper connective pattern that is present, right? So what happens is when the frog looks at the world, it sees a fly over here. The retina shows an forms an inverted image, as we have studied that the real image is that inverted image. So it forms an inverted image at the back of the retina. Once this information is taken to the brain, it is processed, and exactly what the frog's outside world looked like is what he saw, right? So this is what the frog also saw. And once the frog was able to see the fly, it was able to catch the fly and eat it. Right? So this is what it basically means. So in the visual system of frog, the lens projects an inverted visual image onto the retina, and the optic nerve then transfers the image with an additional inversion to the optic tectum. The orderly arrangement of inputs to the tectum is responsible for this transfer. This is the arrangement that we talked about, this anterior, posterior, dorsoventral, right? The neurons in the anterior retina project axons to the posterior tectum. Okay, I'm not going to read this. We have discussed it already, right? What happens in case of an inverted tectum, right? So what was done in Sperry's experiment, this optic nerve of the frog, it was cut, right? And the eye surgically rotated in its socket before the nerve regenerates. Visually guided behavior is aberrant after that, right? So let me show you the complete picture, right? So over here you can see this blue was at the bottom and the yellow was on the top. It is opposite over here now. Over here, towards the nose of this frog was red, but now it is towards the back side, right? No, not no longer towards the nose. So after having surgically removed, this connection actually it this A is still going to P over here as it was before. This P is still going to A as it was before. But now there is some kind of a crisscross that is happening over here. Similarly, ventral is still going to dorsal, dorsal is still going to ventral, but you can see that the crisscross pattern that was earlier present over here, that is lost, right? So what happens is that the kind of image that was being formed in the retina over here, that is not inverted again. Rather, the octoctectum also forms the similar kind of image, right? So how the frog sees the world is like this instead of like this as it should have seen, right? So what this does is it hampers the frog to be able to sight its prey and to eat it, right? So when a fly is present overhead, the frog perceives it as below and vice versa. The inversion of behavioral reflexes result from the connection of regenerating retinal axons to their original targets, even though these connections now transfer an inverted, inappropriate map of the brain to the world. Right? So that is what we studied. So that will be it for part one of the discussion. We'll quickly switch to part two. Yeah, so as we talked about, this will be our final discussion session for learning and memory. So we'll be summing up that particular unit over here today. Yeah, so we'll just begin with this part. So I hope by now you all know the kinds of learning, the kind of memory, the kind of conditioning patterns that are there. What is long-term potentiation? What is spatial memory? What are these regions? Amygdala, hippocampus, right? All this we have studied extensively over the past few weeks, right? So over here, the point that we are going to start with is that spatial memory depends on long-term potentiation in the hippocampus, right? So long-term potentiation is basically that step through which a short-term memory becomes a long-term memory. That step is known as long-term potentiation, right? And over here, which kind of memory is being talked about? Spatial memory. So memory about where things are in your surrounding, right? So long-term potentiation is an experimentally induced change in the synaptic strength produced by strong direct stimulation of neural pathways, right? We have seen that already. To date, a large number of experimental approaches have shown that inhibiting long-term potentiation interferes with spatial memory, right? One spatial memory test uses a pool filled with opaque fluid, which is known as Morris water maze test. It is actually a test for those of you who are going to pursue maybe a um, doctorate or a career in neuroscience or cognitive science, you will probably come across this test. It is also used to, determine the whether the mice are cognitively fit or not. Right. So we'll shortly go through what this test is also exactly. So in this, what happens is to escape from the liquid, a mouse must find a platform which is submerged below the surface of the fluid and is completely hidden from normal view of the mouse. So the animal is released at random locations around the pool and initially encounters the platform by chance. However, in subsequent trials, what happens is the mouse learns to locate the platform and then remembers its position based on spatial contextual cues. So, what are these contextual cues? Um, Even in that circular pool, maybe the platform is always between a cross and a tree, for instance, right? So, a cross is drawn and a tree is drawn that this mouse can see underwater and it is always between that, the platform is between that. So over time, the mouse will learn that, okay, now I have seen the tree, I should go towards the left. There, that is where the platform is. Okay, I have seen the cross, I should go towards the right, right? And if it sees any other cues, it will probably just skip those cues and quickly try to locate this tree or cross so that it can reach to the platform and then escape. So this task requires the hippocampus, right? So there is another version of it. This was dependent on contextual memory. There is another version which is dependent on non-contextual memory. In this non-contextual version of this test, the platform is raised above water. It is not submerged, right? And it is marked with a flag so that it is visible, permitting the mouse to navigate directly to platform using its brain pathways, which do not require an intact hippocampus. So over here, what happens is mouse doesn't need to remember where exactly the platform is or which cues are next to the platform. It just needs to put its head out and see where the platform is, right? So that doesn't require any spatial memory recollection over here. So this is non-contextual version. So what happens is when NMDA receptors are blocked by injection of pharmacological antagonist into the hippocampus, that is something that will go and block this NMDA receptors in the hippocampus. The animal can find the visible platform in the non-contextual version of the task, but it cannot remember the location of the hidden platform in the contextual version of the task, right? So earlier we have seen experiments wherein NMDA receptors were basically silenced, right? So these experiments also suggest that some mechanism involving NMDA receptor in hippocampus, perhaps the long-term potentiation itself, is involved in spatial learning. We also saw that NMDA receptors are required for induction, but not for persistence or maintenance of LTP, right? So, for instance, what would this mean? That if the mouse has already learned where this particular task is, right? During learning, if you are blocking the NMDA receptors, it will not be able to perform the contextual task. But what if it has already learned and after that you are blocking the NMDA receptor, right? So, because it is not responsible for persistence or maintenance of this LTP, it should ideally not affect the mouse memory, right? So similarly, the injection of NMDA receptor blocker into hippocampus after animal has learned a spatial memory task does not inhibit subsequent memory recall for that task, right? That is what was done and that is what was seen also. Yeah. So I think this is delta instead of hash. So we also saw above that PKM delta is required for maintenance of the LTP, but it is not involved in its initial induction. So it comes into picture after the role of NMDA receptors, right? So Todd Sapter and his colleagues have found a corresponding requirement for PKM delta in the persistence of memory. Thus, injection of a pharmacological inhibitor of PKM delta to the hippocampus one day after the animal has been trained on spatial tasks disrupts the memory for that task, right? Which is what is expected also. NMDA receptor blockage at this point will not disrupt the memory, but something that is responsible for maintenance of LTP and not the induction of LTP itself, it will affect at this point also, right? Yeah. So more direct evidence for correlation of memory and LTP come from experiments with mutant mice that have genetic lesions that interfere with this long-term potentiation. One interesting mutation that we will study about is produced by genetic deletion of NR1 subunit of the NMDA receptor itself. Neurons lacking this subunit fail to form functional NMDA receptors, right? So essentially, it is going to somehow affect the formation of LTP or initiation of LTP itself, right? So this is what happens. So these are the mice model that have been built. This is the transgenic mouse line 1, which is homozygous for floxed gene encoded with NR1 subunit of NMDA glutamate receptors, right? So it is homozygous. Both the loci have the same NR1 subunit with the LOXP sites also. And there is this other transgenic mouse line, which is the Cree controlled, wherein Cree is controlled by the CAM-K2 alpha promoter. Cree recombinase is expressed at sufficient levels selectively in the CA1 cells. Right? So what happens is these mice are crossed so that head of this mice will be received, and this progeny will basically have in the CA1 regions a Cree recombinase, which removes genes flagged by the lock sites. So this recom and other one progeny will have no subunit, and the other in the other progeny the recombination will not occur in the cells of the rest of the mouse because Cree recombinase is not expressed, right? So in one it will be expressed, in the other it will not be expressed. So the one in which it will not be expressed and the LOXP site will not be removed, that will have normal NMDA receptors. The one in which it would have expressed, the NR1 site would have been fluxed out, due to which there will be no NMDA receptors in the CA1 neurons, right? So this will be a transgenic mouse trait. So mice with general deletion of this subunit die soon after birth, indicating importance of these receptors for neural function. However, it is also possible to generate lines of mutant mice in which NR1's deletion is restricted to the CA1 pyramidal neurons and occurs only 1 or 2 weeks after birth. Right, so it will have to have some kind of an induction, right? So it is either TET on TET off system, or that doxycycline based system, right? Some treatment based induction probably. These mice survive into adulthood and show a loss of LTP in the Schaffer collateral pathway, which we have studied about before. This disruption is highly localized. Nevertheless, the mutant mice have a serious deficit in the spatial memory. So, even though in these mutant mice, even after this deletion is restricted to the CA1 region only, right? Even after that, although the mice are surviving, the spatial memory is seriously affected, right? What it means is that CA1 region is responsible majorly for the spatial memory tasks, right? So this is that other transgenic mice, right? So over here, it says there is a temporal restriction of gene expression. That is when in time the gene will be expressed, that is restricted. So how it is restricted is there is this TTA mouse line that is taken. It is expressed in the forebrain neurons only, right? It also has a CAMK2 promoter over here. And then there is a TET2 mouse line that is taken, which also has a CAMK2 aspartate region, right? So this mutant form of CAMK2 is under control of TET-O promoter, which is inactive without bound TTA, right? So the progeny of these two mice, what will happen is TTA protein is made and it activates TET-O promoter, leading to transcription of the mutant CAM2, CAMK2 aspartate 286, right? So this is the mice which will have abnormal LTP. When this mouse is fed with doxycycline, which basically suppresses this TTA activity and reduces this TETO signaling, what happens is the binding of doxycycline to TET blocks activation of TETO promoter by TTA and transcription of mutant CAMK2 aspartate 286, and it is shut off. So this basically restores the long-term potentiation to its normal level, right? So this is that extra, the other mouse line, which has only in the CA1 region, it will be affected, and it will also be temporally restricted, right? So, yeah, you can read this also. We have already gone through the system. Also, if you want more, information about how what is the normal transgenes, how are they made, what are these spatially restricted or temporally restricted gene expression patterns, and maybe you can also go through some genetics course or some maybe IVF related course. So that is where these things are also you can revise, right? You might have already read about it, but you can revise it. Then uh, this is what we were talking about, right? So now we know that there are two types of receptors. Two types. One is the wild type, which has the NMDA receptors. One is the mutant, which does not have NMDA receptors, right over here. So you can see that in the wild type, the long-term potentiation curve looks like this, the EPSP for this, and in the mutant, it looks like this, right? So LTP is abolished in mice with NMTA receptor is selectively deleted in CA1 pyramidal neuron by knocking out the NR1 subunit gene. Field EPSPs were recorded in response to Schaffer collateral stimulation, tetanic stimulation at 100 Hz for 1 second, which is shown by this arrow over here, and it caused a large potentiation in wild type mice, but it failed to induce the LTP in the NMDA receptor knockout mutant mice, as you can also see over here, right? Post this, there's this Maze water learning test that we had talked about earlier. So you can see that in case of the wild type, its escape latency is less. Latency meaning how delayed the escape is, right? So with the number of trials, the latency keeps reducing. Now that roughly is happening with the mutant also. The latency, the time taking with each trial is lesser, but you can still see it is significantly higher than that in the wild type mice, right? Yeah. Then there was this other test also which was done, which is the pro-prior test of memory, right? So if you look at the movement patterns, the wild type shows more movement into this quadrant that is marked, while the mutant roams about almost equally in all the quadrants, right? So after the mice have been trained in Morris maze, in the platform is taken away, right? In this probe trial, wild type mice spend a disproportionate amount of time in the quadrant that formerly contained the platform, that is this one, the target quadrant, indicating that they remember the location of the platform even if it is not there anymore, right? But the mutant mice spend an equal amount of time, that is 25%, in all of the quadrants, that is, they perform at chance level, indicating deficient memory over here, right? Uh, right. So, although it is perhaps not surprising that genetic manipulations can impair neuronal function, in some cases, genetic changes can actually enhance both hippocampal LTP and spatial learning and memory. Right? So, how can this be enhanced? It is basically by this HCN based signaling. Right? So, what happens over here is I hope that it will, okay, that will come later. So first, what we talk about is the NR2B subunit. So the one of the first examples of such an enhancement comes from the studies of a mouse mutant that overexpresses the NR2B subunit. So NR1 subunit we were removing, it was causing a disruption of this NMDA receptor. But when NR2B unit, it is not removed, or instead it is overexpressed, what it causes is that more calcium will enter the NMDA from this NMDA receptors. What it will cause is, it is normally present at early stages of development, but it is much more downregulated at an adult hippocampal synapses. The receptors which incorporate this subunit allow more calcium influx than those which do not, right? So in mutant mice that overexpress the NR2B subunit, LTP is enhanced, presumably because of an enhancement of calcium influx. And importantly, learning and memory for several different tasks is also enhanced over here, right? So this is a visual representation of this. You can see the wild type mice and you can see the NR2B transgenic mice. The amplitude of current generated by the NMDA receptors is in response to a brief pulse of glutamate is enhanced, and its time course is also prolonged, as you can see over here, in hippocampal neurons obtained from mice that contain a transgene that expresses higher levels of this NR2B compared to the wild type mice, right? Also, when you look at long-term potentiation, just look at the EPSP, right? Wild type is much lesser after the signal induction, 100 hertz for 1 second, than the transgenic, right? So, long-term potentiation produced by tetanic stimulation of Schaeffer collateral path synapses is greater in transgenic mice than in case of the wild type mice, right? Also, if you look at the Morris water maze learning, spatial learning is enhanced in transgenic mice which have an overexpressed NR2B subunit. As demonstrated in this particular plot, right? This is the latency of wild type, which is higher than the latency of the transgenic mice actually. The rate of learning in a Morris maze is faster in transgenic mice than that in the wild type mice. Also, spatial memory is also enhanced in the transgenic mice as demonstrated in the probe trial test, that same quadrant it is going to or not, right? So, transgenic mice spend more time in the target quadrant, which previously contained the hidden platform, than do the wild type mice. So, they are performing better than the wild type mice also. Thereafter, you can see over here, uh, just a moment. Yes, I'll just reshare the screen. So over here you can see an LTP deficit is seen in hippocampal slices from the transgenic mice which overexpress a constitutively active form of CAMK2, which is this CAMK2 ASP286, right? So the doxycycline, the Tete and doxycycline treated mice that we talked about, right? The expression of this kinase is under the control of TETO and the TTA transcription factor. Four groups of mice were tested. The transgenic mice that are fed doxycycline, which blocks expression of kinase. Transgenic mice without the doxycycline, in which the kinase is expressed. The wild type mice with and without doxycycline. In wild type mice, a 10 hertz tetanus induces LTB and doxycycline has no effect at all. Uh, but in transgenic mice, right, the tetanus fails to induce LTP but causes a small synaptic depression instead, right? So this is the level and it causes a synaptic depression instead of stimulation. In the transgenic mice that are fed doxycycline, the deficit in LTP is reversed. So this is what this spatial memory test looks like. Spatial memory depends on long-term potentiation in the hippocampus, as we were reading about. This is known as the Barnes maze. The effect of kinase on spatial memory was tested in the Barnes maze. Over here, what happens is it consists of platform with 40 holes on its sides, one of which leads to an escape tunnel that allows the mouse to exit the platform. Mouse is placed on the center of the platform. It does not like open well-lit spaces and therefore tries to escape from the platform by finding the hole that leads to the escape tunnel. The most efficient way of learning and remembering the location of the hole and the only way of meeting the criteria for the task by the experimental is by using the distinctive markings over here. You can see on the four walls as cues for hippocampal dependent spatial memory, right? So you can see over here how these mice performed. So the wild type mice, it performed okay. Transgenic mice almost couldn't remember where this tunnel was at all. But once it was again given doxycycline, it could remember again, right? Because the Tete activity was again not present. So, in addition to receiving input through Schaffer collaterals, the CA1 neurons also receive excitatory input from entorhinal cortex, right? The synaptic plasticity at this entorhinal inputs in CA1 may also contribute to spatial learning and memory. Then comes this other example that I was speaking about, the HCN one. right? The HCN1 hyperpolarization activated cation channel is strongly expressed in a very distal dendrites of CA1 neurons, the site of the interrenal inputs. These channels are partially open at the resting potential, which decreases the membrane resistance or RM of the dendrite. This reduces the size of EPSP in response to given excitatory synaptic current and decreases the membrane time constant as well. The net effect is a reduction in spatial and temporal integration of EPSPs in the perforant path. So, if a mice lacks this unit, that is HCN1 unit, it shows marked enhanced temporal summation of EPSPs in the distal dendrites in response to tetanus because of increased membrane time constant. This contributes to large increase in magnitude of LTP at the perforant paths of these synapses, right? So, in contrast, what happens is if the same mice showed relatively little change in EPSPs or LTP at Schaffer collateral synapses, which are formed on or proximal regions of CA1 dendrites, where expression of HCN1 is relatively modest. Importantly, we should note that the mutant mice get smarter and they exhibit a significantly faster rate at which they learn to find hidden platforms in the Morris maze relative to the littermates that express normal levels of the HCN1, right? So, this lack of this particular subunit, like gain of N2B subunit, made these mice more efficient. Similarly, lack of HCN1 subunit also made these mice more efficient. So such experiments support the view that LTP at most distal perforant path inputs to CA1 neurons also contribute to the spatial learning and memory, right? Uh, then a spatial map of the world is basically formed in the hippocampus, right? So it was this basis which has led to a recent Nobel Prize, maybe in the last 10 decades, last 1 decade or so, or in the last 10 years, which has gone to a group which has proven that there are these place cells, place cells and satellite cells in the hippocampus, and it is definitely responsible for the spatial map of the world, right? So that has also been a recent Nobel Prize um, that has been awarded especially in the field of neuroscience. So in 1971, John O'Keefe and John Dostrowski made a remarkable discovery in rats that hippocampus contains a cognitive map of animal spatial environment. How they came to this was an animal's familiarity with particular environment is represented in hippocampus by the firing pattern of population of pyramidal cells which are termed as place cells in the CA3 and CA1 regions. The place cells fires when an animal enters a certain location in a specific environment. And this environment is known as place field. For instance, I will have some place cells which fire only when I go to my house, right? So that is the place field, and those set of place cells will fire which are associated to my recollection of the spatial memory within my house, right? So this is what it looks like. This is the cue card, uh, and the mouse is tracked. The brainwave pattern is also studied, and it is seen that the firing increases as the mouse enters the place cell's optimal zone, right? So, firing pattern of pyramidal cells in hippocampus create an internal representation of animal's location in its surroundings. Electrodes implanted in the hippocampus of the mouse are attached to a recording cable, which is connected to an amplifier attached to a computer-based spike discrimination program. The mouse is placed in a cylinder with an overhead TV camera that transmits to a device that detects the position of the mouse over here. The cylinder also contains a visual cue to orient the animals. Spikes in individual hippocampal pyramidal neurons, which are the place cells, are detected by the spike discrimination program. The firing rate of each of these cells is then plotted as a function of animal's location in the cylinder. This information is then visualized as a two-dimensional map of color-coded firing rates for the cell, from which the cell's place field can be determined. Yellow, orange, red, green, blue, and purple pixels show over here. This is a representation for our understanding, right? So brain might not be, storing it as color-coded structures. These show regions with progressively increasing rates of firing. The place field is the location in space that elicits optimal firing in the cell. Then a population of place field specify the environment. When the animal enters a new place field, new environment, new place fields are formed within minutes, and these are stable for weeks to months. Thus, if one records the electrical activity of a number of place cells, it is possible to predict where the animal is in its environment. In this manner, the hippocampal is thought to constitute a cognitive map of the animal's surrounding. Thereafter, in 2005, Edward and Maybrit Moser, I think it was them who received the Nobel Prize, if I'm not wrong, and their colleagues in Norway discovered the neurons in medial entorhinal cortex whose axons from the perforant pathway to the hippocampus map space in a very different manner from the hippocampal place cells. Instead of firing when the animal is in a unique location like place cells, entorhinal neurons, termed grid cells, fire wherever the animal is at any of the several regularly spaced positions, forming a triangular grid-like array. This grid allows the animal to locate its body with a Cartesian-like external coordinate system that is independent of context, landmarks, and or specific markings. This graded spatial information conveyed by the entorhinal inputs is transformed within the hippocampus to unique spatial locations represented by the firing of the place cells. Right? Thereafter, the mice lacking the NR1 subunit of NMDA receptors. So effectively, they did not have the NMDA receptors. The hippocampal pyramidal neurons still fire in place fields despite the fact that the LTP is blocked. Thus, this form of LTP is not required for transformation of spatial sensory formation into the place fields. However, place fields of hippocampal neurons in the mutant mice are larger and fuzzier than in outline than those in normal animals, right? Thereafter, in a second experiment with mutant mice, late LTP and long-term spatial memory were selectively disrupted by expression of a transgene, which encodes a protein inhibitor of protein kinase A. In these mice, place fields are also formed, but the firing patterns of individual cells are stable only for an hour or so. Thus, late LTP is required not only for formation but also for long-term stabilization of the place fields. Right, so this is a wild type mouse, and this is the mutant mouse. You can see a very uh, different pattern in each of the sessions. While this is showing similar patterns, at least in this region in all of these sessions. So disruption of long-term potentiation alters the stability of place field formation in the hippocampus cells. The color-coded firing rate maps show the place field recorded in 4 successive sessions from a single hippocampal pyramidal neuron in wild-type mouse and from a neuron in the mutant mouse that expresses the persistently active CAMK2. Before each recording session, the animal is taken out of the cylinder and sometime later reintroduced into it. In each of the 4 sessions, the place field of the cell in the wild type animal is stable. In this example, the place cells fires whenever the animal is in the upper right region of the enclosure, as you can see over here. By contrast, place field of cell in the mutant mouse is unstable across all the different sessions, right? Then we come to attention and spatial information, right? So studies in primates have shown that the importance of free prefrontal cortex and modulatory dopaminergic system during attention, right? So the formation of stable place fields in mice require the action of dopamine on D1 or D5 type of receptor, which stimulates adenylyl cyclase, leading to production of CAMP, which is a second messenger, and thereafter, this second messenger, it leads to activation of protein kinase A or PKA, right? So that signaling pathway gets induced. This demonstrates that rather than being a form of implicit memory that is stored and recalled without conscious effort, long-term memory of a stably formed place field requires an animal to attend to its environment, as is the case for explicit memory in humans, right? So, thereafter we come to the discussion that different subregions of hippocampus are required for pattern separation and for pattern completion. So, when we talk about explicit memory, since we are saying that LTP is more similar to, for place cells, the memory is more similar to explicit memory. Let us recall what it is, right? So, explicit memory is used to store facts, which is the semantic memory, and episodes, which is the episodic memory. Such successful storage and recall of explicit memory requires the ability to distinguish between two closely related images, episodes, or spatial configurations, which is known as pattern separation. And second, the explicit memory can also use partial cues to retrieve previously stored memories by filling in an incomplete pattern based on pre-existing knowledge, which is an ability which is known as pattern completion, right? So these are two different distinct abilities that we have. A variety of cell physiological and computational studies, beginning with the theoretical work of David Marr in 1970s, suggest that the pattern completion depends on the recurrent connections between CA3 pyramidal cells, and the pattern separation depends on the direct projection from the entorhinal cortex to the dentate gyrus. Right, the dentate's role in pattern separation was suggested because the number of granule cells in the dentate far exceed the number of CA3 or CA1 pyramidal neurons. The importance of LTP between CA3 neurons is seen in the studies performed on mice in which the NMDA glutamate receptor is selectively deleted in CA3 neurons. These mice experience a selective loss of LTP at the recurrent synapses between CA3 neurons and with no change in LTP at the mossy fiber synapses onto the CA3 neurons or at the Schaffer collateral synapses between the CA3 and CA1 neurons. Despite this deficit being present, these mice show normal learning and memory in the water maze. However, when the mice are asked to find a hidden platform with fewer than normal number of spatial cues, their performance is impaired. This indicates that LTP at recurrent synapses between CA3 neurons is important for pattern completion, right? Similarly, mechanisms of pattern separation has been examined in mice in which the critical subunit of NMDA receptor is selectively deleted from granule neurons in dentate gyrus, or in which the neural activity of the granule is blocked. Mutant mice lacking the functional NMDA receptor in the dentate gyrus have difficulty distinguishing between two similar contents. That is, the mice cannot perform pattern separation. Okay. Thereafter, we come to a topic that a lot of people are not aware of. So we say that neurons are the only cells that do not regenerate. So recent studies, which are not that recent also, so we know since some time now that there are certain groups of neurons. Not all neurons can regenerate, but there are actually some groups of neurons that can be formed later in life also, right? So, one of the most unexpected findings in neuroscience has been the realization that neurogenesis is not limited to early stages of development. New neurons continue to be born throughout adulthood and become incorporated into the neural circuits. However, adult neurogenesis is limited to two types of neurons in the brain in two brain regions. One is the inhibitory granules in the olfactory bulb, and the excitatory granule neurons of the dentate gyrus. Right? When neurogenesis in the dentate is blocked, either by X-ray irradiation of specific brain regions or administration of chemical that interferes with the DNA synthesis, there is a viable effect on learning and memory. Some forms of hippocampal dependent memory are impaired, whereas other appear intact. However, recent exciting results showed that pattern separation specifically requires the participation of adult born new granule neurons, right? So, to learn a lot of things, we do require new neurons to be formed, right? Moreover, procedures that stimulate neurogenesis enhance the ability of a mouse to perform pattern separation during a contextual memory task, which requires an animal to distinguish between closely related environments. Also, memory depends on long-term depression of synaptic transmission. Right, so this is a concept that is totally antagonist to the LTP, right? This is LTD. If synaptic connections could only be enhanced and never attenuated, synaptic transmission might rapidly saturate. The strength of the synaptic connection might reach a point beyond which further enhancement is not possible. Yet, individuals are able to learn and store new memories throughout a lifetime. This paradox led to the suggestion that neurons must have mechanisms to downregulate synaptic function to counteract LTP also, right? So, in fact, such an inhibitory mechanism, which is termed as long-term depression or LTD, was first discovered in the cerebellum, where it is important for motor learning. Since then, LTD has also been characterized at a number of synapses within the hippocampus. Whereas LTP is typically induced by brief high frequency tetanus, induction of LTD requires prolonged periods of low frequency synaptic stimulation, such as stimulation at 1 Hz for 15 minutes. LTD can also be induced by pairing protocol, which is in which an EPSP is evoked after a postsynaptic cell fires an action potential. This suggests an anti-Hebbian learning rule. So Hebbian learning rules we have studied. This is an anti-Hebbian learning rule, that is synapses do not contribute to firing of cell are weakened, right? Over there, what it said was synapses that fire together, they get strengthened and they get linked together, right? Like LTP, number of molecules and synaptic molecular and synaptic mechanisms are available to produce LTD. Surprisingly, many of the forms of LTD also require activation of the same receptors involved in LTP, namely the NMDA receptors. Moreover, like LTP, LTT is thought to require calcium influx through the NMDA receptors into the postsynaptic neurons. So, you can see over here, NMDA receptors are required for long-term memory, long-term depression also, right? So, we'll see how it is different from their expression patterns, right? That in depression also the same receptor is involved. So prolonged long-term frequency stimulation, that is, for instance, 1 Hz for 15 minutes of Schaffer collateral fibers produces a long-term decrease in size of the field EPSP in hippocampal CA1 region, a decrease that outlasts the period of stimulation or control. The diagram shows that long-term depression LTD results from removal of AMPA receptors from the postsynaptic membrane by endocytosis. This LTT is blocked when the 1 Hz stimulation is given in the presence of APV over here to block the NMDA receptors. So again, if NMDA receptors are blocked, then LTD is also blocked. So no LTP, no LTD. Yeah. Uh, Long-term depression requires a protein dephosphorylation. Protein phosphatase 2A is required for this process. The plots compare LTD in CA1 region of wild-type mice and transgenic mice that express a protein that inhibits the phosphoprotein phosphatase 2A, right? Transgene expression is under control of the TTA system. In the absence of doxycycline, when the protein inhibitor is expressed, induction of LTD is inhibited in the left plot, as you can see. When the inhibitor protein expression is turned off by administering the doxycycline, a normal sized LTD is induced, as you can see in the right plot. The inhibition of phosphatase 2A alters behavioral flexibility. Transgenic mice expressing phosphatase inhibitor learned the location of submerged platform in Morris maze at the same time as wild-type mice. Learning is measured by the daily decrease in the path length the mice traverse as they search for the platform during training. At the end of day 10, the platform is moved to a new heading location. Mice are rested. Now, the transcending mice require significantly longer path lengths to find the platform on the first day of the retesting. When transgene expression is turned off with doxycycline, the transgenic mice display normal learning on all phases of the test. Compared to the high frequency stimulation used to induce LTP, the low frequency tetanus used to induce LTD produces relatively modest postsynaptic depolarization and thus much less effective at relieving the magnesium block of the NMDA receptors. As a result, increase in postsynaptic calcium concentration is much smaller than observed during induction of LTP. This lower concentration is thought to be insufficient to activate CAMK2, which is the enzyme which is implicated in LTP, right? Compared to high frequency stimulation, rather LTD is thought to result from the activation of calcium dependent phosphatase calcineurin, an enzyme complex that has higher affinity for calcium compared to CAMK2. So if calcium is there in less volume, it will probably go to this calcineurin instead of CAMK2, and LTD will be the outcome of that, right? Activated calcineurin triggers a signaling cascade that activates still other phosphatases that dephosphorylate a number of proteins, including the uh, uh, glutamate, GLUA1, which is also known as GLUR1, which is a subunit of the AMPA receptor. Okay. In addition to activating phosphatases, LTD induction protocol also increases the phosphorylation of GLUA2 or GLUR2 subunit of AMPA receptor by protein kinase C. The combined effects of dephosphorylation of GLUA1 and phosphorylation of GLUA2 trigger the endocytosis of AMPA receptors, reducing the number of receptors in the postsynaptic membrane and thus reducing the size of the EPSPs that are seen, right? Also, distinct forms of LTP can be induced through activation of metabotropic glutamate receptors. Such forms do not require activation of phosphatases but depend on activation of mitogen activated protein MAP kinase signaling pathways. These types of LTD lead to reduction in synaptic transmission through a decrease in glutamate release from Schaffer collateral terminals as well as through alterations in the trafficking of AMPA receptors in the postsynaptic cells. Okay, we'll come to this. So epigenetic changes in chromatin structure are also important for long-term synaptic plasticity and learning as well as memory. Right, what it means, what are epigenetic changes? These are basically changes which happen or modifications on the proteins that generally happen, right? These modifications could be like acetylations, dephosphorylations, attaching of phosphates, methylation, etc., right? As is the case with long-term implicit memory, long-term explicit memory storage also requires covalent alterations in the chromatin structure. Such changes are termed as epigenetic changes, right? So it is not at the gene level, it is post processing of the protein, right? One of the epigenetic regulations involves acetylation of histone proteins. Now, this is something we have all heard about. The protein component of the nucleosome repeat unit of the chromatin. Over here, what happens is CREB binds to camp recognition element 3 promoter that recruits a transcriptional co-regulator, the CREB binding protein or CBP, which functions as a histone acetylase, right? Acetylation of N-terminal tails of histone by CPP disrupts repressive chromatin structure and opens up the promoter region of the target genes, allowing the binding of RNA polymerase to initiate the transcription, right? Thereafter, what happens is this is how acetylation looks like, right? So, if the gene has to be expressed over here, post the formation of this protein, the histone is acetylated. So, what happens? Phosphorylation of CREB1 in following induction of late phase of LTP in hippocampus recruits binding of the CBP, that is Cre binding protein, to promoter region of the target genes. In turn, CBP acetylates certain positively charged lysine residues on N-terminal tails of the histone proteins, which form nucleosome of the unit of the chromatin. Acetylation loosens the binding of negatively charged DNA to histones, allowing transcription to proceed and resulting in the late phase of LTP. Just a moment.
Hello, ma'am. I guess your mic is off, so we are not hearing you. Well, I will not hearing you. I'm not sure if others are hearing. Am I audible?
Yes, yes, you are actually audible. Mm, I had turned off the mic because of the coughing, right? So I am sharing the screen again. Yeah, I hope I'm audible now, right? So we were talking about these epigenetic changes, right? So one of the epigenetic changes that we had talked about was acetylation. And what happens? That is a normal case that we have talked about. But sometimes there could be mutations in one of these processes, right? So mutations in CBP, for instance, the Cre binding protein, it leads to a syndrome, which is a rare condition, which is known as Rubinstein-Taibi syndrome, right? It is characterized by a mental retardation and skeletal abnormalities as well. What happens in this syndrome is it is modeled by mice by insertion of a truncated non-functional form of CBP into the mouse genome. These mice show a reduction in the histone acetylation and impairment in the late phase of the LTP that is associated with the deficit in certain forms of hippocampal dependent long-term memory also, right? Whereas uh, defects in histone acetylation result in memory impairment. Enhancement of histone acetylation using inhibitors of histone deacetylase can actually enhance memory. Okay, they might have some off-target effects also, but they have actually been shown to enhance memory also. So, uh, a second form of chromatin modulation that has been implicated in the learning and memory involves DNA methylation, right? It is one of the most common epigenetic change that is generally studied, right? Methylation. So certain cytosine bases that precede guanine base in DNA form CPG sites which can be methylated by DNA methyl transferases. DNA methylation recruits methyl CPG binding proteins that in turn bind transcriptional core processor complexes that inhibit the gene expression in part because these complexes contain these HDACs, which are the uh, histone deacetylating enzymes, right? The mutations in, sorry, uh, oh, this is the same slide again, right? Yeah, maybe I went back instead of forward. Yeah. So this is the other one that we were talking about. Gene repression could be caused by either deacetylated chromatin or methylated DNA, right? Induction of LTP also leads to changes in methylation of certain cytosine bases that precede guanine nucleotides or CPG sites in DNA through the action of DNA methyltransferases, DNMT. This recruits methyl CPG, uh, methylated CPG binding proteins, which in turn recruit histone deacetylases or HDACs over here, which leading to decrease in histone acetylation, which together with CREP2 represses transcription, right? A second form of chromatin modulation that has been implicated in learning and memory involves DNA methylation. Certain cytosine bases that precede guanine bases in DNA CPG islands can be methylated by DNA methyltransferases. DNA methylation recruits methyl CPG binding proteins that in turn bind the transcriptional core processor complexes that inhibit gene expression, in part because of these complexes contain HD, ACs, right? DNA methylation is thought to be of particular importance during early development when it helps initially to determine the cell fate and maintain cell identity. Evidence that DNA methylation is important for proper development of the human nervous system is also illustrated by the fact that mutations in methyl CPG binding protein 2 underlie Rett syndrome, which is a pervasive neurodevelopmental disorder. The DNA methylation play a huge role in learning and memory in the adults as well. Contextual fear conditioning in mice causes increase in methylation of gene encoding, the neurotropin BDNF, right? BDNF you might have heard in association with heart distress, right? Or cardiac distress in CA1 region of the hippocampus, which leads to changes in the BDNF expression. Injection of pharmacological inhibitors of DNA methylation to CA1 both inhibits the change in BNF expression and impairs fear memory 24 hours after conditioning, right? So, concluding this unit about learning and memory, uh, the final question that we actually began with, the first question and the final question was that are there molecular building blocks for learning, right? So at a uh, more systemic level, we have always studied that, okay, this region of the brain might be responsible for this region of the brain might be responsible for this is how learning happens, right? But are there actually molecular building blocks for this to happen, right? And do we understand that? There might be, but do we understand them, right? So we have talked about it somewhat during this particular course of understanding learning and memory, be it the Cree based responses or Krebs based cycles or calcium influxes, and how these various different receptors, NMDA receptors, and others are getting involved, right? So all that is molecular level itself. So, the three key findings have emerged from cellular studies of implicit as well as explicit memory storage, right? One, the molecular mechanisms of some associative forms of synaptic plasticity are based on those of non-associative forms in the same cell. This suggests that there may be molecular building blocks for synaptic plasticity. Simpler forms of plasticity might serve as components for more complex mechanisms, such as joint recruitment of presynaptic and postsynaptic mechanisms of plasticity. Second, the molecular mechanisms of elementary forms of associative learning, both implicit and explicit, are similar. Two synaptic mechanisms from for memory storage we have considered. One is activity dependent presynaptic facilitation for storage storing implicit memory, and associative long term potentiation for storing explicit memory, right? These seem to derive from the associative properties of specific proteins. Example, the responsiveness of adenylyl cyclase or NMDA receptor when two independent signals are simultaneously present, right? Finally, we have also seen that despite clear differences, implicit and explicit memory storage seem to rely on elements of common multi-component genetics which involving protein kinase A, PK, CREB, epigenetic changes in chromatin structure that convert liable short-term memory to long-term memory. Moreover, these mechanisms of synaptic plasticity do not operate in isolation. Rather, they are embedded in distributed neural circuits that have considerable computational power and thus can add substantial complexity to the action of these individual cells. So, overall, one thing is that, for instance, we have just seen that NMDA, the biggest answer for this is NMDA receptors are involved in long-term potentiation also and long-term depression of those same memories also, right? So it is probably the same molecular players, but what are the other interacting players over there is what is defining whether what kind of a learning process is happening, right? Whether it is inhibitory, excitatory, what is happening, right? So that is what is C. So that will be it for this session. If there are any questions, please feel free to ask. Also, till now, I have not really planned what exactly I'll be, explaining in this extra session for the last session that I have to take. Just a minute, I'll stop sharing. So if there is something particular that you would want me to discuss about, maybe I can prepare according to that. Maybe I can discuss that particular part in detail. You can write to me also if you want to. Um, it is Arveen K21 at iitk.ac.in. If you are writing to me, please do it latest by Friday because then I'll prepare for the class for next Monday, which will be our final lesson. Also, for this, if you have something right now, you can also let me know. Yes, most probably what I'll be discussing is some neurodevelopment and no, okay. There was one thing that was left that I had thought that if I have time, I will share. Those are basically techniques, diagnostic level techniques for the brain, right? Or which are applicable for humans also and neuroscience in general also. So if nothing from your end, that is what I'll be uh, explaining in the next session. So that will be all from my side. Thank you for the patience for joining. I'll just stop the recording. Good night, everybody.